The enzyme RNA polymerase unwinds the double helix structure of DNA. It then scans the DNA and once it reaches a promoter region, binds onto it, attaching itself to the template strand through the help of transcription factors. Transcription factors are accessory proteins that regulate the rate at which transcription occurs and can either be activators, which speed up transcription, or repressors, which slow down transcription. The RNA polymerase II then creates a transcription bubble by breaking the hydrogen bonds between the complementary base pairs and separating the two strands of DNA. A RNA-DNA helix structure is created when the RNA polymerase creates a hydrogen bond between the mRNA strand and the DNA strand.
RNA polymerase travels down the template strand, creating complementary base pairs to the nucleotides it travels over. By using base pair rules, theoretically, RNA polymerase should create a cytosine for every guanine and adenine for every thymine, and vice versa. This, however, isn't true, as uracil is created whenever RNA polymerase encounters an adenine on the template strand. Uracil is used as this is less energetically expensive to create. The RNA polymerase travels down the template strand from the 3' end to the 5' end, creating an mRNA molecule that is 5' to 3'. The 5' end and 3' end are used to add a sense of directionality to the DNA and also shows that a phosphate group attaches to the 5' end while a hydroxyl attaches to the 3' end. While the RNA polymerase is traveling down the DNA strand, it simultaneously zips the DNA back up and will only have 10 to 20 nucleotides exposed at any single given time.
The final stage is termination. As the RNA polymerase is adding complementary nucleotides to the 3' end of mRNA, it reaches a section of the DNA which is the terminated region. This signals that the mRNA transcript is complete and the DNA and mRNA strand break the hydrogen bond disassociating. This then also results in the joining of both template and coding DNA strands. The mRNA created in transcription is called pre-mRNA and is unable to be used in translation. This pre-mRNA includes regions which are called introns and exons. Intron splicing uses spliceosomes to cut out the introns, which are non-coding parts of the gene. These are then excised, leaving only the exons, which are the coding parts of the gene. Spliceosomes are made up of proteins and RNA, making it a complex structure. A 5' cap, which is a modified guanine, is added to the 5' end, while a poly-A tail consisting of 50 to 250 adenines is added to the 3' end, allowing for the mRNA strand to reach maturity. The final mRNA strand leads through pores in the nuclear membrane where it travels to the cytoplasm for the next step in polypeptide synthesis to occur. Translation is a step in which the mRNA strand created in transcription is used to create a polypeptide or chain of amino acids. The nucleotides in the mRNA are in groups of 3 called codons. For translation to occur, we need 3 important structures. These structures are mRNA, tRNA aka transfer RNA, and ribosomes. Just like transcription, translation also has 3 stages, which are also called initiation, elongation, and termination. For initiation to occur, a small ribosomal subunit will bind to the mRNA strand at the methylated guanine cap on the 5' end upstream of the mRNA strand. Each amino acid which creates the polypeptide chain is brought by a tRNA molecule. The tRNA molecule structure is quite important as it holds both an amino acid, which joins to create a polypeptide, and a specific triplet of nucleotides, which are complementary to the codons in the mRNA and are called anticodons. For translation to occur, the first amino acid to start this process is methionine, which is coded for by the codon AUG and the anticodon UAC. In most cases, this is the first amino acid produced, and the codon AUG is responsible for it. The factor that decides which amino acid is brought forth by the tRNA molecule is decided by the order in which the nucleotides in the codon are in. This can also be read through a codon table or chart. A large ribosomal subunit then attaches the mRNA strand, tRNA molecule, and small ribosomal subunit, creating the peptidyl and the aminoacyl sites. These are also called A and P sites. The first tRNA occupies the P site and the new tRNA molecule enters through the A site. The second tRNA molecule is complementary to the next set of codons on the mRNA strand, and the methionine from the first tRNA molecule joins to the threonine through a peptide bond. The tRNA from the P site then leaves through the E site or exit site, and the ribosome moves along the mRNA strand. the tRNA molecule occupying the A site moves to the P site, allowing for a new tRNA molecule to enter. The ribosome then moves along reading the next set of codons, and the next tRNA molecule carrying the complementary anticodons and amino acid are brought into the A site. In this case, it will be phenylalanine. The process of the ribosome moving down the mRNA, reading codons, corresponding tRNA molecules entering through the A site, the growing peptide chain being continuously transferred to the A site from the P site, and the tRNA molecules from the P site leaving through the E site are the main process of elongation. Termination occurs when a stop codon is present in the A site. This brings forth the release factor instead of a tRNA molecule. This results in the disassociating of the ribosome, mRNA strand, release factor, and tRNA molecule. This newly formed protein is released to the E site. Before it is used in the body, it may or may not need to be modified before it is ready to function.